Good morning students, welcome to my channel Digital Generation. This is a graph which represents the progress of different components of food digestion in human alimentary canal. The parts P, Q, R, S, T represents different parts of the alimentary canal. Study the graph and answer the following question. Identify the parts P, R and S. We have to identify the parts P, R and S. These all parts are the parts of alimentary canal of human digestion system. Here is a key. This blue line represents carbohydrate digestion. This red line represents protein digestion. And this green line represents fat digestion. So from here we can see that carbohydrate digestion starts from P. We know that carbohydrate digestion starts from mouth because of the presence of enzyme ptyalin or amylase in saliva. So P must be mouth. From R, the digestion of proteins takes place. We know that the digestion of protein takes place from stomach. So R must be stomach. And emulsification of fat is also taking place in stomach due to the presence of gastric lipase enzyme. So the part P is mouth because here carbohydrate digestion has taken place but no protein digestion has taken place, and we know that protein digestion does not take place in mouth but protein digestion takes place in stomach. So P is mouth. Q is the esophagus, so we will write here mouth, then it will be easy for us. And R is the stomach because from here the protein digestion has started. In this graph the carbohydrate digestion has become stable. We know that in stomach there is no carbohydrate digestion and emulsification of fat is taking place because of the presence of an enzyme gastric lipase in stomach. So R is stomach. And S is duodenum of small intestine because the emulsification of fat has taken place because of the presence of bile juice which is poured into the duodenum, and thus fat digestion has taken place. Carbohydrate digestion has restarted because of the presence of beta amylase enzyme in duodenum which has been secreted by the pancreatic juice and has been poured into the duodenum. This beta amylase enzyme converts the remaining starch into maltose. So S is duodenum. And T is obviously ileum, where all the digestion has been completed and all the foods has reached to its end product. Next question, explain protein digestion in the graph. From here, this is the graph of protein digestion. We will explain it according to the graph. Protein digestion is taking place from stomach. R is stomach, and in stomach there is hydrochloric acid and makes the medium acidic, and there are two enzymes in stomach known as pepsin and renin which acts in acidic medium and thus converts complicated protein into proteoses and peptones. There from stomach it is transported to duodenum, and in duodenum pancreatic juice is poured in, and thus this pancreatic juice contains enzymes trypsin and chymotrypsin which acts in alkaline medium. The bile juice is also poured in duodenum which makes the medium alkaline. So the partially digested proteins or proteoses and peptones which are formed in stomach are further digested and gives polypeptides in the presence of enzyme trypsin and chymotrypsin. And now in the form of polypeptides the protein is transported to ileum. In ileum complete digestion of protein and all other foods also complete digestion of food takes place, but here we are considering the protein. So in ileum complete digestion of protein takes place by the presence of various polypeptidase enzymes secreted by the intestinal juice. These polypeptidase enzymes converts polypeptides into amino acid, and in ileum the amino acid is formed, and amino acid is the end product of protein, and from ileum this amino acid is absorbed by villi present in the ileum of small intestine which is richly supplied with blood vessels, and thus absorption of protein takes place in ileum. Next question, which part among P, Q, R, S and T receives the secretion of digestive gland? The part duodenum receives the secretion of digestive gland. Name two digestive glands and their secretion. The two digestive glands are liver which secretes bile juice and pancreas which secretes pancreatic juice. This pancreatic juice and bile juice are poured into duodenum. This bile makes the medium alkaline, and this bile also helps in emulsification of fat. So the emulsification of fat is the highest at duodenum, and in ileum it is completed. And the pancreatic juice gives various enzymes like beta amylase enzymes which converts remaining starch into maltose, and the enzymes like trypsin and chymotrypsins which converts partially digested proteins or proteases or peptones into polypeptides. And fat emulsification or fat digestion is taking place by lipase enzyme. So the two digestive glands are liver and pancreas. Liver which secretes bile, and pancreas which secretes pancreatic juice. And in ileum there is secretion of various types of enzymes. Lipase which converts fat globules into fatty acid and glycerol. Various polypeptidase enzymes which converts polypeptides into amino acids, and beta amylase enzymes which converts maltose into glucose, fructose or galactose. Thus all the complete digestion is taking place in ileum. State two differences between pepsin and trypsin enzyme. While studying the graph, we have come across about the two enzymes pepsin and trypsin. Pepsin enzyme is present in stomach, or it is produced in stomach, and trypsin enzyme. Both these enzymes helps in digestion of protein. The complicated proteins are digested into simpler forms by this enzyme. This pepsin enzyme is present in stomach, and it converts complicated proteins into proteases and peptones. This trypsin enzyme is produced in pancreas, and it converts these proteases and peptones into polypeptides. Thus, pepsin acts in acidic medium because it is produced in stomach, and stomach produces hydrochloric acid. So, pepsin enzyme acts in acidic medium, and trypsin enzyme acts in alkaline medium because bile is alkaline in nature and bile is poured in duodenum. So, the medium becomes alkaline. Thus, trypsin enzyme acts in alkaline medium. Next question. Name 3 disaccharides and 3 monosaccharides. Disaccharides are maltose, lactose and sucrose. These are the 3 types of disaccharides. Monosaccharides: glucose, fructose and galactose. These are monosaccharides. So what is starch? Starch is polysaccharides. Starch is polysaccharide. When starch is taken in, then it is polysaccharide. So it is converted into disaccharide, either maltose, lactose or sucrose, which starts from mouth. And these disaccharides is converted into monosaccharides in ileum in the form of glucose, fructose and galactose. So in ileum there is complete digestion of starch also. Thank you for watching my video, and if you find my video useful, please do like and share my video and subscribe my channel to get the latest update of my videos. And if you have any doubt regarding this topic, comment below. I will try to answer as early as possible.